This is something I found on Pinterest and I thought it was funny. Now, after this short break, after this, let's talk about what happens at the ends of the chromosomes. So the bacteria don't have this problem. They have a circular chromosome. When they finish replication, the two polymerases move in the opposite direction and meet somewhere in the area that's directly opposite the origin of replication, and the ligase will fuse this connection. But eukaryotic cells don't have circular DNA. Their DNA is linear and has been packed into chromosomes. As a result, if you understand how DNA replication works, you immediately realize that there's a problem at the end. Because once the Okazaki fragments have been removed, once the Okazaki primers have been removed here, there is no place at the very, very tip for yet another DNA polymerase to come in and fill in that place, because the synthesis, it needs a primer and can't synthesize without a primer. It can't fill out from the 3' to 5' direction. It has to go 5' to 3'. So this problem of the lagging strand was ignored for a very long time, and it was only in the early 90s that that the existence of these special structures at the end of the chromosome has been appreciated. So in this case, they're called telomeres, and they, in this chromosomal squash of I think human chromosomes, maybe mouse, they have been labeled in white. So all of the DNA has been labeled in blue, and then because the ends of the chromosomes have a very special nucleotide pattern, why we'll see in a second, you can create a special probe that will bind to them. Remember southern and northern blot? Well, you can do this also without breaking up the cells, but just on non-degraded chromosomes, and you can use this hybridization to label the special areas of the chromosome called telomeres in a different color. It's an approach called FISH, fluorescent in situ hybridization. So hybridization is DNA and probe binding. In situ means inside either a cell or, in this case, a nucleus, a squash nucleus. And fluorescent is because these guys, instead of being labeled with radioactivity, like you do in a southern or northern blot, have been labeled with a fluorescent dye that has been attached in a particular way to the nucleotides of the probe. So the enzyme that maintains the ends of chromosomes is called a telomerase. It's a curious enzyme. It's a complex between RNA and proteins, and this is what it does. It caps the chromosomes with short repetitive sequences, thus you can make probes against them and recognize them pretty easily. This is a typical GGGTT. This is a very typical telomere sequence that telomerase synthesizes. Oops. And one thing I want to point out here is because telomerase is not a DNA polymerase. It's a reverse transcriptase. It uses its RNA template to synthesize the DNA in a 3' to 5' direction. So you can see here that in a 3' to 5' direction, it uses its internal template, an RNA that's been incorporated inside the telomerase, and it's called telomerase RNA molecule, right here, to use it as a template. So it has base paired right here, and then will synthesize, reverse transcribe from the RNA into DNA, the telomere. Then it will translocate further and make some more. As a result, because it has created this long stretch of 5' to 3' RNA in the direction that's usually alien to all DNA polymerases, an RNA primer can be synthesized by a primase, and then a proper normal DNA polymerase can extend this primer and fill in the gap. Then the RNA primer will be removed, and the very, very end of a typical telomere is protected by these single-stranded 3' end binding proteins. So telomerase, as I said, is an unusual combination of RNA and protein. Another unusual combination of RNA and protein is the ribosome. And you can see, so it has two parts. It has the TERT, which is telomerase reverse transcriptase. This is a group of enzymes, a group of proteins bind together, and there's a particular active site there that can catalyze the reverse transcription reaction. As well, the other part of a telomerase is RNA. It has a short template that is used by the TERT then as it's used by the TERT in order to synthesize, to extend the telomeric sequence. And if you look at the blot, if you want to look at how TERT enzyme activity or telomerase enzyme activity, you guys can see that in yeast, if you just give telomerase a short stretch of DNA and some DNTPs, it will start making these sort of short stretches and then extend them and extending them and extending them to make them bigger and bigger and bigger. So in this case, this is called, this is a telomerase activity assay. It's sort of a combination of a southern blot and DNA gel and enzyme activity assay. And you can see the activity of a particular telomerase by how many bands it can synthesize in a given amount of time. In this case, all of these, it was fed radioactive nucleotides, so it has created radioactive DNA. And telomere maintenance is very important. So most of our cells have very low telomere activity. They don't care quite so much about preserving their telomeres. However, the cells of our germline, aka the sperm cells and the egg cells, have very, very high activity and very long telomere lengths because it is their job to give pristine, unaltered DNA to the next generation, because it is the fusion of sperm and egg that will create the next living organism. Stem cells, which are cells responsible for replenishing many tissues of our body, also have very high telomerase activity. Well, you guys can see here that normal somatic cells don't. However, telomerase is reactivated in many cancers and so has become a target for various anti-cancer therapies. If you can inhibit the telomerase activity in cancer cells, you can destabilize their chromosomes and cause the cells to die. And these are just food for thought. And I think I pretty much answered them for you. All right, this concludes our DNA replication lecture. The next lecture I will try to post by Monday. And next time we're going to be talking about various DNA technologies that scientists employ. The reason we're going to talk about them ahead of talking about transcription and translation is because it will make it easier to understand how scientists work with DNA and RNA and proteins. Have a good Memorial Day weekend. Bye.